Hello everyone, welcome to this very important session, Target Prelims 2024 Science and Technology. So in this session what we are going to do is pick up some of the most important topics that we have seen in the last 1 year from the field of science and technology, and I'm going to discuss with you some of the very crucial questions that we can expect or topics that we can expect in Prelims 2024. So let's begin with question number 1. Question number 1 says, consider the following statements regarding neutrinos. First, neutrinos are electrically neutral and have very small mass. Second, neutrinos interact weakly with matter, making them difficult to detect. And third, neutrinos are produced in nuclear reactions such as those occurring in the sun and nuclear reactors. So now when it comes to neutrinos, and neutrinos very often you will see that they have been in the news, and here the context that we are picking is one of these articles that we saw in the newspaper. With neutrinos, scientists observe our galaxy in a whole new way. Now what are neutrinos? Neutrinos basically are fundamental particles that are very key part of the standard model of particle physics. And when it comes to these neutrinos, we know that neutrinos, they are neutral. They do not carry any electric charge. That's why it is something, that's why in the name itself it reflects the term neutral. So that's why they are derived from this term neutral, and hence they are called as neutrinos. Similarly, when it comes to their mass, they have a very small mass, almost negligible mass, much less than even the mass of an electron. Although the mass is not zero, their exact mass is still unknown, but it is known to be very small. So that's why there are two things that we know, that one, they are electrically neutral, and second, they have a very small mass. So first says neutrinos are electrically neutral and they have a very small mass, which is perfectly correct. Then the second statement says neutrinos interact weakly with matter, making them difficult to detect. Now this also is correct. When it comes to neutrinos, there are three different types of neutrinos, and each of them is associated with a corresponding charge, something called as lepton. So there are basically three types of neutrinos: electron neutrino, muon neutrino, and tau neutrino. Alright, so just know this, that there are electron neutrinos, muon neutrinos, and tau neutrinos. These are the three types of neutrinos. Now these neutrinos, they interact with matter via a very weak force, and weak force basically is one of the four fundamental forces of nature. Now this weak interaction makes them incredibly difficult to detect because they can pass through ordinary matter almost undisturbed. So that's why what we see is that detecting them becomes very difficult, because they interact very weakly with matter, and that's why we see that it's very difficult to detect them. There have been experiments around the world where we have been trying to detect them, but it has been very difficult for us to do that. Now coming to the sources of neutrinos, neutrinos basically are produced in various different types of nuclear reactions. Whether it comes to the nuclear fusion reaction that is happening inside the sun, which produces neutrinos which can be called as solar neutrinos as well. Alright, we can call them as solar neutrinos, or in case of supernova, which is kind of an explosion that happens at the center of a star or a massive star which is about to die, supernova also produces a lot of neutrinos. And then we see that there are a lot of cosmic ray interactions with the Earth's atmosphere also, which lead to the production of these neutrinos. And at the same time, the nuclear reactors, they also become source of neutrinos. So apart from that, as I just told you, that cosmic ray interactions with the Earth's atmosphere, this also becomes one of the reasons. So 1, 2, 3 and 4. all these can be said to be the various sources of neutrinos. So neutrinos are produced in nuclear reactions, which is correct, such as those occurring in the sun and nuclear reactors. So that's why this is perfectly fine. So all these three statements given here are correct. So correct answer will be 1, 2 and 3. So moving on to the next question. The next question is about a very important mission called as the Euclid mission. So the question says, consider the following statements regarding the Euclid mission. First, the Euclid mission is led by NASA to study the sun and its effect on the solar system. Second, one of the primary objectives of the Euclid mission is to understand the nature of dark matter and dark energy. And third is, Euclid spacecraft is equipped with instruments that capture data in both visible and near field wavelengths. Now again, when it comes to this particular question of Euclid mission has been in the news, and this is regarding exploration of the dark universe. Now when we say dark universe, what it basically means is that it is going to study dark energy and dark matter. Dark energy and dark matter. Alright, so this is what it is going to study. Now in this particular scenario, when it comes to the Euclid mission, there have been a lot of news articles. You'll see that at least there have been 4 or 5 different news articles in the last 1 year which are related to the Euclid mission, and that is why Euclid mission becomes very crucial for your examination this year. So first is, the Euclid mission is led by NASA to study the sun and its effects on the solar system. Now this is not correct. It has not been led by NASA, but rather by the European Space Agency. And that's why the first statement is not correct. The first statement is not correct because it is led by the European Space Agency. So that's why first statement is wrong. The second statement, one of the primary objectives of the Euclid mission is to understand the nature of dark energy and dark matter. This is what we discussed, that this is going to study the dark universe. So that's why the correct statement, the second statement is correct. And then the third statement, Euclid spacecraft is equipped with instruments that capture data in both visible and infrared wavelengths. Now this also is correct because there are different equipments. For example, there is an equipment called as the visible imaging channel. There is one that is called as visible imaging channel. Visible imaging channel. So this is one of those instruments which will do the interactions, which will capture the interactions in the visible range. And then there is near infrared spectrometer and photometer, which is called as NISP or NISP. Near infrared, right? Near infrared spectrometer and photometer, or also called as NISP in short. So this basically is what captures data in the visible light. So overall, that's why we see that there are all in the near infrared. So this NISP is for near infrared and this VIC is for visible. So that's why this is also correct. So correct answer will be 2 and 3 only, that is B. Right, so moving on to the next question. Question number 3. Question number 3 says, with reference to the Laser Interferometer Gravitational Wave Observatory or LIGO, consider the following statements. First, LIGO is designed to detect gravitational waves using large interferometers arranged in an L shape. Second, currently there are 2 LIGO setups located in the United States. Third, LIGO India will be constructed in the Hingoli district of Maharashtra and is scheduled to begin operations in 2030. And fourth, LIGO India project will be managed solely by ISRO. Now, coming to why this article, because we have seen that it has been under discussion that India is going to start the operations of LIGO, or that is LIGO India, which we also are calling as Indigo. So it is also being called as Indigo sometimes. This is basically the LIGO arrangement that we are building in India. So the announcement had happened a long time back, almost in 2016 approximately. But now we see that the budget also has been approved last year, and that's why it is back in the news. So let's discuss. So first statement is about whether the arrangement detects gravitational waves using L-shaped arrangement. So you can see that this is correct, that you have a kind of an L-shaped arrangement in which this kind of a detection is done. So that's why this particular is a correct statement. Second, there are two LIGO setups in the USA. This is correct. One is there in Louisiana and the other one is in Washington. So that's why this is also correct. So one is in Louisiana and the second one is in Washington. Second one is in Washington. So these are the two that are there in the United States. The third statement is also correct, that we see that LIGO India is being constructed in Hingoli in Maharashtra, and we are expecting that the operations will begin by 2030. And then lastly, the last statement is not correct. Alright, last statement is not correct, that it is not solely managed by ISRO. So this is not correct. So correct answer will be 1, 2 and 3. Because LIGO India will be run by 3 Indian institutions, and again in collaboration with LIGO Laboratory that is operated by the California Institute of Technology and the Massachusetts Institute of Technology, that is MIT. So that's why this statement, the fourth statement, is not correct. Moving on to the next question. With reference to ISRO's Small Satellite Launch Vehicle or SSLV, consider the following statements. First is SSLV is capable of launching multiple satellites in different orbits during a single mission. The development of SSLV aims to reduce the turnaround time for the launch operations. SSLV uses solid propulsion system for all its stages. And then lastly, SSLV can be assembled and launched within a week. So these are the 4 statements that have been given here. Now, first statement about SSLV. First coming to the context of why exactly are we talking about it, because last year it was in the news that ISRO would transfer the SSLV to the private sector, and then there was another news later in the month of September that there are 23 companies which have shown interest in getting this SSLV technology or using this technology. Alright, so SSLV. What is SSLV? SSLV is Small Satellite Launch Vehicle. Now when we say Small Satellite Launch Vehicle, it means that it is capable of launching satellites which have a total weight of less than 500 kg. Less than 500 kg. Because when we say that a satellite is a small satellite, the meaning is that it weighs less than 500 kg. So that is what SSLV has been designed for. And this SSLV is being managed by NSIL. It is being managed by NSIL, that is New Space India Limited, and that's why we see that there are these things that become crucial. Now first statement here is correct, that the propulsion module of when it comes to the module that we have here, it is capable, right? This SSLV is capable of launching multiple satellites in different orbits during a single mission. This is correct. The development of SSLV aims to reduce the turnaround time for launch operations. This is also perfectly correct. This is also a correct statement, that this is basically what we want, that the turnaround time for the operation should be reduced and we should be able to get all this done very quickly. Uses solid propulsion system for all its stages. This is also correct, that there are 3 stages in total. There are 3 stages in total, and all these 3 stages are solid. So we are using solid fuel for all these three stages. But just to add to it, there is another module towards the end which is called as the velocity trimming module. And this velocity trimming module uses a liquid fuel. But it is not counted as a stage, it is just there with the satellite so that we can get the transfer of the satellite done to the required orbit. So for that we need a liquid fuel, which is velocity trimming module. Otherwise all these three stages are solid stage, so this is also correct. The fourth statement, it can be assembled and launched within a week. This is correct. I have taken this from official statement that was made by ISRO, where they have claimed that within a week they would be able to be ready with the launch of SSLV. So that's why all these four statements given here are correct statements. Moving on to the next question. With reference to India's Chandrayaan 3 mission, consider the following statements. Now, the first is propulsion module carries the shape payload to study Earth's spectral and polarimetric measurements from lunar orbit. The lander is equipped with instruments to measure lunar thermal conductivity, seismic activity, and plasma density. Third, the rover's primary objective is to conduct laser-induced breakdown spectrometry to understand the lunar atmosphere. I understand that some of the words or some of the names that you see here you may not know, and that is precisely what I wanted here, because we know the basic information about Chandrayaan 3. You know that there is a lander called as Vikram, there is a rover which is called as Pragya. So these are two parts of the Chandrayaan 3 mission. So when it comes to Chandrayaan 3 mission, let me just break down some of the things, and of course it has been in the news multiple number of times. But here we are discussing about the payloads. What are we carrying in this satellite? So you have the lander which has been named as Vikram, and you have the rover which has been named as Pragya. Right? So this is what we have, and there are all these different types of instruments that we have on board. So first statement talks about the propulsion module. So propulsion module, as you can see, it does carry the spectropolarimetry of habitable planet Earth or shape. So this propulsion module, it carries this shape payload, and this payload is designed to study Earth's spectral and polarimetric measurements from the lunar orbit, and that's why this statement accurately reflects the mission's detail. Secondly, when it comes to the second statement says that the lander is equipped with instruments to measure lunar thermal conductivity, seismic activity, and plasma density. Now this also is correct, because if you look at all these different payloads that you have, first is Rambha to measure the near surface plasma density and the changes of it with time. So which is what we saw here, plasma density. Second, you have chaste, and this is to carry the measurements of thermal properties of lunar surface near the polar region. This is what we see here, thermal conductivity. And then the third is ILSA, and this is for measuring the seismicity around the landing site. So that's why these are the three lander payloads, and this is also, that's why, correct. And then after that you have the third statement saying rover's primary objective is to conduct laser induced breakdown spectrometry to understand the lunar atmosphere. Now in this particular scenario, when it comes to the rover payloads that we have, as you can see that there are these two payloads that we have here. One is APXS and the second one is LIBS. Now in this particular scenario, when it comes to these payloads, understand that what exactly are they doing. First is to derive the chemical composition and intermineralogical composition to further enhance the understanding of the lunar surface, and secondly to determine the elemental composition of the lunar soil rocks around the lunar landing site. So when you look at statement number 3, the rover's primary objective is not to understand the lunar atmosphere, right? It is not about the lunar atmosphere. Correct? It is not about the lunar atmosphere. It is basically what? First, when you look at the first payload that we have here, the first payload or even the second payload, these are intended to derive the elemental composition of the lunar surface in the vicinity of the landing site. And the focus basically is on the analysis of the lunar soil and the rocks rather than studying the atmosphere of the moon. So that's why first and second statements are correct. The third statement is not correct. So that's why the correct answer will be B, 1 and 2 only. Now in this context, one more thing that becomes important is to also understand about the lunar south pole. The lunar south pole is where we have tried, and you'll see that there are other missions also. We'll discuss about some of the other missions where we have been trying to get to the lunar south pole. So understand why lunar south pole. What is the importance of lunar south pole? First thing is that there is presence of water ice near the lunar south pole. The lunar or the moon's south pole is believed to have significant deposits of water ice, primarily in the permanently shadowed craters of the moon. And these ice deposits, they can be used to produce water for even human consumption, or even oxygen for breathing, and maybe hydrogen for required fuel. So that's why the presence of water ice near the lunar south pole is one of the primary reasons. Then also resource utilization wise, if we see, the availability of water ice is something that makes lunar south pole very attractive site for future lunar bases as well, and also for long-term human presence if we want. And that's why it also enables, you can say in a manner, an in situ resource utilization. Also reduces the need for transporting water from the earth to the moon if at all we have a base there. At the same time, the lunar south pole also has areas which have been in permanent shadow for billions of years, preserving very ancient geological materials as well. And studying these areas could provide us insight about the Earth's solar system, about the early solar system and history of the moon, and even perhaps the Earth. Then apart from that, the cold traps that we have at the south pole may also contain a lot of volatile compounds beside water ice. For example, carbon dioxide or methane, ammonia, all these things could also be present, and these are very important for understanding of the moon's composition and evolution. So that's why there are all these things that we have to understand. So early, it can give us an insight about the early solar system, and then other volatile compounds like carbon dioxide or methane or ammonia. All these things could also be present. So that's why there are all these so we can get a better understanding about the moon's composition and even the evolution of the moon. So that's why all these things. So that's why from Chandrayaan 3 perspective, there are these things that we should rather know. Moving on to the next question, question number 6. Which of the following statements is or are correct regarding astronomical phenomena? First is about gravitational lensing. Gravitational lensing occurs when a massive object causes the light from a more distant object to bend and magnify. Second, a supernova is an explosion of a star that significantly increases its brightness and can outshine entire galaxies for a short period. Third, the cosmic microwave background is the thermal radiation left over from the Big Bang and provides evidences for the expansion of the universe. So here we are talking about 3 different phenomena which are related to the understanding of the universe. So first is gravitational lensing. Now gravitational lensing is a phenomena where a very massive object like even a galaxy or clusters of galaxies, they bend the light from a more distant object and it creates multiple images, magnifications, or even some kind of arcs of the background objects. So something like this, that from distant galaxies, as you can see that because of galaxy clusters, what is happening is that the path of light is getting bent. Alright, the path of the light is getting bent, and this bending of the light is called as gravitational lensing. So as you can see that one kind of some kind of a lens has been formed here, so that is called as gravitational lensing. So the first statement, that's why, is correct. The second statement is about a supernova. Now supernova is indeed the explosion of a massive star that increases the brightness of this star drastically, and that's why very often we see that it can outshine the entire galaxy for a very brief period of time. So it is nothing but an explosion at the center of a massive star which is about to die. So this also is correct. The third statement is about cosmic microwave background. Now this cosmic microwave background is a kind of an afterglow radiation from the Big Bang, and it provides very crucial evidence for the Big Bang theory. It is said to be one of the crucial evidences that supports the Big Bang theory, and also the kind of expansion of the universe that the Big Bang theory talks about. So that's why this is also correct. So all these three statements given here are correct. So correct answer will be D. Moving on to question number 7, again a very important mission from India's perspective, and it is about the Gaganyaan mission. Consider the following statements about the Gaganyaan mission. First, it is an ISRO mission aimed at sending Indian astronauts to low Earth orbit for short duration. Second, the mission involves a launch vehicle called LVM-3, which is a 3-stage rocket. Third, the orbital module includes a gynoid called as Vyomitra that is capable of monitoring capsule conditions and performing certain tasks. The fourth is Indian government has set up a coordinating body for Gaganyaan called as the Human Space Flight Center. Now, when it comes to ISRO, ISRO has successfully tested the Gaganyaan service module propulsion system at the ISRO propulsion complex, and this was in the news. Alright, this is something that was in the news, and this test basically was involving 5 different motors or 5 different liquid motors that we had. So there were 5 different liquid motors of 400 tons. So 400 newtons. So 400 newtons liquid. I'll just write the name just in case they ask something related to it. Liquid apogee motor. This test was done. Alright, and there were different control systems also which were tested. Now in this particular system, what we see is that this module, this system that we are talking about, this system which is also called as the SMPS system. Alright, this is also called as the SMPS system. Now this SMPS system basically is designed for orbital injection and circularization, and also for onboard controlling and deboost maneuvering of this entire system. Now this test that was done, it basically demonstrates the integrated performance of the propulsion system which was done with the these kind of engines like the LAM systems. So overall what we see is that the tests are happening with regard to the Gaganyaan system, and there have been all these kind of tests, and especially these configurations, they pave the way for the further test and to demonstrate both the nominal and off-nominal mission scenarios. So when it comes to the Gaganyaan mission, we know that it aims to demonstrate the human spaceflight capability by launching 3-member crew to a 400 kilometer orbit for a 3-day mission, and then also trying to get all these people or the 4 astronomers also back to the Earth. So in this particular scenario, when it comes to the mission, the Gaganyaan mission, the first, it is a mission aimed at sending Indian astronauts to low Earth orbit. So Indian astronauts to low Earth orbit for a short duration is correct. So that's why statement number 1 is correct. Just important thing here is that you have to read each and every part of your statement. Everything given in the statements, you should not miss any of it. Alright. So that's the first statement. The second statement, the mission involves a launch vehicle called as the LVM-3, which is a 3-stage rocket. So LVM-3, this is also correct. LVM-3 or Launch Vehicle Mark 3, which also in many times, in many cases, is also called as GSLV Mark 3. So GSLV Mark 3 or LVM-3, both basically are the same. Just different names, otherwise they are the same. So it is a 3-stage rocket, which is correct. Then the third is the orbital module includes, now it is not the orbital module, alright, it is the crew module. It is the crew module that will have a gynoid. So when we say gynoid basically means that it is a female form of a robot. So we use the terms android and gynoid many a times. Although androids many a times is also used for any human form of a robot, but gynoid specially is used for the female form of a robot, which is Mitra here. So this part is correct, but it is not the orbital module. So that's why the third statement is not correct, and correct answer will be 1 and 2 only. Let's look at question number 8 now. With reference to the X-ray polarimeter satellite or XPOSAT and the PSLV orbital experimental module, that is POM3, consider the following statements. First statement is XPOSAT is designed to measure the polarization of X-rays in the energy band of 0.8 to 15 kilo electron volt. Then XSPECT payload on XPOSAT is responsible for long-term spectral and temporal studies of cosmic X-rays. And third, POEM3 is an initiative by ISRO to carry payloads that can perform in-orbit scientific experiments and technology demonstrations. And then lastly, both POLIX and XSPECT payloads on XPOSAT cover entirely non-overlapping energy bands for their respective measurements. Now in this particular question, let's first try to understand what exactly is there. So first thing, why exactly are we talking about? We are talking about this particular very important mission of ISRO, where we have been talking about XPOSAT and has been in the news multiple number of times. So when it comes to XPOSAT, it measures the polarization of X-rays in the energy band of 8 to 30 kilo electron volt. 8 to 30 kilo electron volt, and that's why this statement given here is not correct. And then secondly, when it comes to the XSPECT payload on XPOSAT, this is basically responsible for the long-term spectral and temporal studies of the cosmic X-rays. So this is perfectly correct, that this is exactly what it is supposed to do. Even the third statement given here is also correct, because the POEM3 is used for carrying payloads that can perform the scientific experiment, that is in orbit scientific experiments and all the technological demonstrations. So that is exactly why it has been designed by ISRO. So that's why second and third statements are correct, first is not correct, and fourth statement again is incorrect, because they do have an overlapping energy band where both can carry out these measurements, which allows for combined data analysis. So that's why saying that they have non-overlapping energy bands is wrong. So statement number 2 and 3, these are the two statements which are correct. So these are the two payloads that we are talking about. So now when it comes to the PSLV orbital experimental module or POM3, it is an initiative by ISRO to utilize the spent fourth stage of the PSLV as an orbital platform for conducting scientific experiments and for technological demonstrations. Now understand this, that when it comes to this POM3, why exactly is this important? What exactly does it do and why is it important? So first thing is that when it comes to PSLVs, if you know, PSLVs, they have 4 stages. They have 4 stages, and often the fourth stage does not really have a lot of use. It is just required for placing the satellite to the required orbit. That's it. Now, this is where, with the help of this poem 3, we want to utilize the fourth stage. Alright, so after basically the primary payload, once the primary payload is deployed, the fourth stage of the PSLV, which is usually discarded, it is something that can be repurposed as an experimental platform. And that's why this innovative approach, it makes the use of otherwise unused resources, and it tries to maximize the utility of the launch. So that's why this becomes very, very crucial. At the same time, it also carries scientific payloads and the instruments that can perform experiments in this particular orbit. That's why these can actually include a lot of different technological demonstrations or scientific research, and various different types of investigations also can be done with the help of this stage. At the same time, this particular stage, when converted to poem, is basically equipped with solar panels for power generation as well, communication systems also, and at the same time they can help in necessary support infrastructure to also maintain the payloads and the experiments in this particular orbit. So that's why the main objective of this poem 3 is to provide a cost-effective and efficient way to conduct the experiments in the space, and also tries to enable the researchers to test new kind of technologies and also gather scientific data without the need of a dedicated satellite. So that's why this is very, very crucial that we are able to use the fourth stage itself as an experimental module. This is exactly what this poem 3 is all about. And then when it comes to the XPOSAT, these are the various scientific goals that we have of XPOSAT. You can pause for a minute, you can read it through, because this can be important for this year's examination, because multiple number of times we have seen that this has been in the news. Coming to the 9th question. Consider the following statements regarding the Luna 25 mission. First, the primary objective of the Luna 25 mission is to execute a pinpoint landing near the moon's south pole to conduct geological samples and data. Geological samples and data. Second, the mission is expected to aid in finding water or its building blocks to support the establishment of human colonies on the moon. Third, Luna 25 was launched using Falcon 9 rocket from the Kennedy Space Center. Fourth, the mission will last for 1 year, during which it will utilize its robotic arm, that is spectrometers and imaging systems, to collect and transmit data back to Earth. Now, when it comes to this Luna 25 mission, it has been again in the news last year. First, because of its launch, and second also because it did not succeed as expected. Now, Luna 25 mission, it aims to achieve a precise landing near the moon's south pole, a region which is very significant in the scientific interest for the potential geological insights, as we just discussed in a couple of in one question, just a couple of questions earlier. Now, this mission, it was supposed to collect geological samples to collect this unexplored area, and also had to find water or the building blocks to support the future colonies on moon. So that's why the first and the second statements, these are correct. Now, the third statement is not correct, because here we have used a Soyuz 2 frigate rocket. Alright, so just remember the name that what we have used here. It is the Soyuz 2 rocket, which is also called as Soyuz 2 frigate rocket. So this has been actually used, and the mission is basically supposed to last for a year, which is also correct as given here in the fourth statement, and during which it was supposed to gather the samples of with the help of its robotic arm, use the spectrometers for the data collection, take images and transmit all this information back to the Earth. So that's why the first statement is correct. The second statement is also correct. The third statement is not correct. Alright, the third statement is not correct, and then the fourth statement again is correct. So that's why when it comes to this particular question, the correct answer will be A, that is 1, 2 and 4. Moving on, question number 10, and again a very important question because of Aditya L1 mission. Consider the following statements regarding the payloads of Aditya L1 mission. First, the visible emission line coronagraph is designed to study the outer layer of the sun's atmosphere. The solar ultraviolet imaging telescope will observe the photosphere and chromosphere of the sun in ultraviolet wavelengths. The plasma analyzer package for Aditya will investigate the solar wind and its interactions with the Earth's magnetosphere. So these are the three statements given. Now, when it comes to the payloads that we have on Aditya L1, there are all these payloads that we have. So you have the suit, the Velk, the Papa, the Solex, then you have Helios and Aspects. So these are all these, and obviously magnetometer. So these are the payloads that we have on Aditya L1. So there are 7 payloads altogether that we have here. Now, just to give you a brief about what the role of all these various payloads are. First, the visible emission line coronagraph. This basically is to study the solar corona. What is corona? The outermost part of the sun's atmosphere. So to study the outermost part of the sun's atmosphere and to understand the dynamics and the origin of the coronal mass ejections, and at the same time their impact that we have on the solar weather. So that's why it will try to observe the corona in the visible and the infrared wavelengths to try and measure the temperature, the velocity and the density of the corona. So that's why this is what the Velk is supposed to do. So just understand that what is it going to do? It is going to study the corona, right? It is supposed to study the corona and understand the coronal mass ejections. Understand the coronal mass ejections and the impact of coronal mass ejections on the space weather. So that's why this is basically what it is supposed to do. It will try to measure the velocity, the temperature and the density of the corona, and the studies will be done in the visible and the infrared wavelengths. So this is correct, to study the outer layer of the sun's atmosphere. Then after that we have the solar ultraviolet imaging telescope or SUIT. Now the role of this SUIT is to image the solar photosphere and the chromosphere in the ultraviolet spectrum. So in this particular scenario we are talking about the visible and the infrared, right? Visible and the infrared. Whereas in case of SUIT we are talking about the near ultraviolet spectrum. It will do its study near the ultraviolet spectrum, right? So it will basically provide full disk, or it actually has already provided full disk images of the sun in various different ultraviolet wavelengths, and this is what will help us try and understand the spatial and the temporal variations that happen in the solar atmosphere and study the solar irradiation in the variability of the irradiation of the sun. So this is exactly what it is supposed to do, and that's why we see that there were pictures also that were published with different filters that were used by the SUIT, where it was able to give us images about the solar flares or the sunspots and all these kind of things. So that's why this is also correct. The second statement is also correct. Then after that we have the Aditya Solar Wind Particle Experiment as well, as you can see here, what we call as aspects. Now the study, this basically the role here is to study the wind, the solar wind's particle flux, the composition and the energy distribution. So largely this is, just remember that largely it is regarding study of the solar winds. study of the solar winds, and this will basically try to analyze the charged particles which are emitted by the sun and their interactions with the Earth's magnetosphere, and that's why it will try to provide insight into the space weather phenomena as well. And then we have the plasma analyzer package for Aditya. Now the role here is to study the solar wind and the interaction with the interplanetary medium as well. It will try to measure the properties of the solar wind plasma, such as the density, the velocity, the temperature, and it will try to contribute to the understanding of the heliospheric environment as well. So this is basically what it is supposed to do. So it will basically try to understand the interaction, alright, interaction of the solar wind with the interplanetary medium. So investigates the solar wind and its interaction with the Earth's magnetosphere, which is correct. So that's why all these three statements given here are correct. Then apart from that, there are others as well. You have the solar low energy X-ray spectrometer, or called as SOLEX. Now the role here is to observe the X-ray flares. The X-ray flares from the sun, and it will try to measure the intensity and the energy spectrum of the low energy X-rays, what we also call as soft X-rays, so which are being emitted by the sun, and it will try to help us understand and study the heating mechanism of the solar corona and the dynamics of the solar flare. So that's why this also becomes very, very crucial. And then when we have the Helios, this is high energy L1 orbiting X-ray spectrometer, Helios. Now this basically is trying to monitor the high energy X-rays. Alright, this is basically for high energy X-rays. High energy X-rays, what we also sometimes call as hard X-rays. So this was about soft X-rays and this is about hard X-rays. So now in this particular scenario, it will try to provide the data on the energetic processes which are occurring in the solar corona, and it will try to contribute to the study of the solar flares and the solar mass ejections as well. Then apart from that, we have the magnetometer. So magnetometer basically it will try to measure the magnetic field variations in the interplanetary space. The magnetometer will also try to help and understand the magnetic field's influence on the charged particles in the solar wind and their interaction with the Earth's magnetosphere as well. So that is basically, you can say it in simple terms, it is about study of the magnetic interactions. Alright, study of the magnetic interactions. So this is what we are doing. So understand this because Aditya L1 has been in the news so often, there are chances that they can ask you a question based on any one of all these payloads, because all these payloads also they have been in the news. There has been one news about the Helios, there has been one news about a suit, there has been one news about Velk, there has been one news about Papa, there has been one news about magnetometer. So there have been all these things, they actually have been in the news. So that's why there are high chances that they can ask you a question based on that. So these were 10 questions which were based on space technology. Now coming to some of the questions related to the energy sector. The first question here is question number 11, that is, consider the following statements regarding nuclear fusion. Now why are we talking about nuclear fusion? Because nuclear fusion has been in the news constantly because of one reason or the other. Especially with regard to the breakthrough that we have seen in nuclear fusion in the last couple of years, we have seen that it has been in the news. First in December 2022 and then again in August 2023, it has come back in the news. So let's look at the statements here. The first statement is nuclear fusion occurs when the nuclei of two light atoms combine to form a heavier nucleus, releasing energy in the process. This is correct. This is exactly what happens, that we get enormous amount of energy when two lightweight nuclei, especially of deuterium and tritium, both of which are what? Both of them are isotopes of hydrogen, right? What are these? These are isotopes of hydrogen, right? So what happens is that they would interact, they would combine, and while combining they become helium. So hydrogen combines to become helium while releasing a lot of energy, huge amount of energy. So that's why first statement is correct, that nuclear fusion involves a combination of two lightweight nuclei to release energy in the process while forming heavier nucleus. Second statement, nuclear fusion is the process that powers the sun and other stars where hydrogen nuclei fuse to form helium. This is perfectly correct, that we see that the source of all the heat and light of the sun and all the other stars is basically nuclear fusion. So that's why this is also correct. The third statement says magnetic confinement and inertial confinement are two primary approaches used to achieve controlled nuclear fusion on Earth. This is also correct, that these are two ways in which we try to get to the conditions that are required for nuclear fusion to occur. Otherwise nuclear fusion wouldn't occur if these conditions did not prevail, because there are very extreme conditions which are required. For example, we need extremely high temperature, we need extremely high temperature, we need extremely high pressure, we need extremely strong magnetic fields at times. This is what you have inside the sun on any other star, right? So that's why these kind of temperatures, pressures, etc., getting them is not that easy. So we have to use certain approaches, certain techniques to be able to do that. So one is called as magnetic confinement. If you have heard of ITER, International Thermonuclear Experimental Reactor, or the new plant that has been built in Japan, all these basically are using magnetic confinement. And inertial confinement, magnetic confinement is where we are using very strong magnetic fields to ensure that nuclear fusion could occur. And inertial confinement is where we use lasers, where we make the use of lasers to confine the plasma so that nuclear reactions could occur. And this has been used in the US. It has been used in the US, where they have been able to get a breakthrough. That is, they were able to generate more energy as compared to what they have spent in the reaction. So that's why this is also correct. Then the fourth statement, deuterium and tritium are the isotopes commonly used in experimental nuclear fusion reactions on the Earth. This is perfectly correct. This is what we discussed. So correct answer will be 1, 2, 3 and 4. As you can see, deuterium and tritium, these are the ones which are being used. Moving on, question number 12. Consider the following statements about advanced battery technologies and fuel cells. Now very often you will see that in the context of electric vehicles or in general the developments in the battery technology, we see that very often these are things which are in the news. So let's discuss that. So first is about lithium ion polymer cell. So lithium ion polymer cell, which are very often used in the smartphones, which is fine, they are used in the smartphones. Lithium metal oxides, they basically serve as cathodes. They serve as cathodes, and graphite basically is not the cathode. Alright, so graphite basically is the anode. It is the anode. So that's why, not the other way around. So that's why the first statement given here is not correct. The second statement, solid state batteries typically have lower energy densities than lithium ion batteries, but they are safer due to the elimination of flammable liquid electrolytes. Now when it comes to solid state batteries typically have higher energy densities than the lithium ion batteries because of the use of solid electrolytes, which can also enhance the safety. So this is fine, that they are safer. Alright, they are safer because we are not using flammable liquid electrolytes, but instead we are using solid electrolytes. We use solid electrolytes here, and that's why they are safer. So but it has a higher energy density, so that's why this is wrong. Third statement is about sodium ion batteries. So sodium ion batteries, they are less expensive due to the abundance of sodium, but they generally have low energy densities and they have shorter lifespan as compared to the lithium ion batteries. So sodium ion batteries offer a potential cost advantage. This is fine because of abundance of sodium over lithium ion batteries due to the abundance and low cost of sodium. This is correct, but the problem is that they have low energy density. The problem with them is that they have low energy density, so that's why this becomes, and at the same time they have shorter lifespan. They have shorter lifespan, so these things you should know. Then the fourth one is about fuel cell. Fuel cells convert chemical energy directly into electrical energy through a chemical reaction with oxygen or another oxidizing agent. Now this is perfectly fine. This is perfectly correct that this is exactly what happens, and this also makes them suitable for continuous power supply. Applications where, for example, where we do not need recharging, where we do not want recharging, in these kind of cells, fuel cells can, that's why, become crucial. So that's why 3 and 4, these two statements are correct, and the first and the second statements are not correct. So sorry, it had to be which of the following are incorrect. Pardon the mistake. So first and the second statements, these are incorrect. Moving on to the 13th question. Consider the following statements regarding the criticality of nuclear reactors. Now, when it comes to nuclear reactor, this word criticality is often very, very important, because you'll see that for any nuclear reactor, criticality becomes crucial, because once criticality is reached, then only you can begin with the reaction. So for example, recently in the month of December 2023, the Kakrapar 4 reactor that India has, this has now achieved criticality. Alright, now it has been able to attain criticality. So that's why in December it was in the news. So what is criticality? So when we say that a nuclear reactor has reached criticality, it is when it now achieves a self-sustaining chain reaction. Alright, a self-sustaining, when it reaches a self-sustaining chain reaction, meaning that the chain reaction can go on in a continuous manner, in a sustainable manner. If a reactor does not reach criticality, if it is subcritical, then the reactions will not happen for a very long time, they will stop. And if it becomes supercritical, meaning that if it goes beyond criticality, then in that case it can lead to an uncontrolled chain reaction, which can lead to accidents, or what basically becomes the source of any atom bomb. So if you control the chain reaction, it is critical and it gives you electricity. If you don't control the chain reaction, it can become supercritical and it can lead to nuclear accidents, which can be very, very dangerous. So let's look at the statements. First statement, a nuclear reactor reaches criticality when it achieves self-sustaining chain reaction, which we just discussed, so this is fine. When a nuclear reactor is critical, the rate of fission reaction is constant. Now this is also correct, that the fission reaction is constant, so that's why it can keep working for a long time. And subcritical reactors can still produce power and are used in some and are used in some power plants. This basically is not correct. Alright, this is not correct, because when it comes to being subcritical, when it becomes subcritical, means that this is where you will not have enough. Correct, you will not have enough and you will not be able to sustain with the chain reaction. So that's why this is not correct. So correct answer will be 1 and 2. Subcritical meaning that less than criticality, you do not have enough neutrons to carry on with the nuclear reaction. So that is why this is wrong. Now moving on to some of the questions related to biotechnology. Which of the following statements about circulating cell-free DNA or cfDNA are correct? First is cfDNA is fragmented DNA that circulates freely in the bloodstream. Second, cfDNA can be used in prenatal testing to determine the genetic conditions of the fetus. Third, cfDNA is exclusively found in cancer patients. Now, cfDNA or circulating free cell DNA is the type of DNA that is found freely circulating in the bloodstream. So this is correct, that it is found freely circulating in the bloodstream. So we have seen that there is this news, cell-free DNA promises to transform how we find diseases. So this was in the news. So as you can see that this is the kind of DNA that is there in the bloodstream outside, freely circulating in the bloodstream. Now when it comes to this cfDNA, understand this, that cfDNA is fragmented DNA that is circulating freely in the bloodstream. So what happens is that it consists of small fragments of DNA that are released into the bloodstream from the cells which are undergoing apoptosis. All right, apoptosis meaning the programming, you can say programming of the cell death. All right, so while they are going apoptosis, all right, while a cell undergoes apoptosis, this is when these DNA fragments are released into the bloodstream. At the same time, any other cellular process, for example, even necrosis, even if the cell is undergoing necrosis. So in case of necrosis, basically, which is an uncontrolled cell death, you can say, this is basically a programmed cell death, and necrosis is an uncontrolled cell death. All right. Now in this particular scenario, what we understand is, or any other cellular process, these kind of fragments, they are typically short fragments. These fragments are short fragments having like 150 to 200 base pairs in the length, and they are found in both the healthy individuals and also with both different types of medical conditions. All right, so that's why they can be found in healthy individuals also, and they can be found in medical conditions, different medical conditions as well. Now the second statement talks about the prenatal testing to determine genetic conditions of the fetus. Now in pregnant women, the cfDNA from the fetus, it can be found in the mother's bloodstream, and that's why this fetal cfDNA, it can be analyzed to detect certain genetic conditions and chromosomal abnormalities as well. For example, something like a Down syndrome or Edward syndrome, these kind of things actually can be found. And now today, that's why we see that there are non-invasive prenatal testing, where commonly we see that cfDNA analysis is done, which provides a safer alternative to the invasive procedures, like the different kinds of procedures that are used in finding this. So that's why cfDNA actually can be used in the prenatal testing to determine the genetic conditions in the fetus. Then the third statement, cfDNA exclusively found in the cancer patient, this is incorrect, because while we do know that cfDNA is elevated, it's higher in number in the cancer patients due to the high rate of cell turnover and the death, but it is not exclusive to them. cfDNA is also present in the healthy individuals, and it can be found in various different conditions such as inflammation, trauma, autoimmune diseases, or even during pregnancy, as we just discussed. So we see that in oncology, the analysis of the cfDNA very often is referred to as liquid biopsy. All right, these terms sometimes again they are in the news, liquid biopsy. All right, they are used in liquid biopsy. It can help in early detection of cancer and monitoring and the treatment and the response, and also trying to detect the residual disease if at all. So that's why we see that liquid biopsy is something that is used in early detection of cancer. Early cancer detection. So not exclusively, it is found, alright, cfDNA is found but not exclusively. So correct answer will be 1 and 2 only. Now coming to question number 15, consider the following statements regarding somatic genetic mutations. Now what are somatic genetic mutations? They basically refer to the alterations in the DNA that occur in the somatic cells. What are somatic cells? Somatic cells are all the cells in our body which are non-reproductive in nature. Meaning that if you set aside sperm cells, egg cells, etc., then all the other cells of the body which are non-reproductive in nature, they are called as somatic cells. Now these mutations that happen in the somatic cells, they happen after the birth and they can actually occur during an individual's lifetime. They are not really inherited from the parents, but they arise due to some of the errors that can happen in the DNA replication or the environmental factors or even exposure to some of the mutagens. So there are all these reasons why this kind of genetic mutation might happen. Now let's look at the statements that we have. The first statement: somatic genetic mutations occur before birth and are inherited from the parents. Now again, as we just discussed, that they are not inherited from the parents, but rather they happen while somebody is while the development of a person is happening. So that's why it is incorrect, that the mutations occur after the birth, during the development, and they are not inherited from the parents. So that's why the first statement is incorrect. The second statement says somatic mutations can sometimes render a cell fitter, leading to the formation of tumors. Now this is correct, that somatic mutations can actually lead to the formation of tumors by making some of the cells fitter than the others, and hence it can lead to tumor formation. The third statement: recent advancements in cell sequencing technologies have significantly increased our understanding of somatic genetic variations. This is a general statement and this is correct, that the advancements in cell sequencing technologies, they have indeed increased our understanding of the somatic genetic variants, and we have better understanding of how they function and what they can lead to. So we see that there are better understandings that we have today. And then the fourth statement is also correct. The SMAHT network is basically to study the somatic mosaicism that we have, and they also have received significant funding from the US government. So that's why this is actually also correct, and this basically is a network that is aiming to study the somatic mutation. So that's why 2, 3 and 4, these are the three statements which are correct. The next question: consider the following statements regarding the differences between DNA and RNA. Now RNA, for one reason or the other, very often has been in the news. So let's look at the statements. The first is DNA contains the sugar deoxyribose, whereas RNA contains the sugar ribose. This is correct. That's why RNA and DNA, RNA is ribonucleic acid, whereas DNA is deoxyribonucleic acid. So that's why the first statement is correct. The second statement: DNA is typically double-stranded while RNA usually is single-stranded. Right? So this is also correct, that we have a double helical structure in a DNA like this, whereas RNA is a single helical structure like this. So that's why this is also correct. The third statement: uracil is found in DNA but thymine is found in RNA. Now this is opposite, because you have adenine, guanine, cytosine and thymine. These are the four which are found in DNA, but instead of thymine you have uracil. The rest three being the same, uracil is found in RNA. So that's why the third statement is wrong. DNA is more stable than RNA under alkaline conditions. this actually is also correct, that RNA under moist conditions, what we see is that RNA is very less stable, but in drier conditions RNA can be more stable. So this is correct, that DNA is more stable than RNA in moist conditions, but in dry conditions RNA also can be very stable. So this has been in the news. Scientists recover RNA to resurrect extinct Tasmanian tiger and species. So that is the reason why we are talking about this, because they have done the RNA analysis and they have been able to find RNA which came from very early organisms as well. Now when it comes to DNA, we have found DNA for very ancient organisms as well, but RNA for ancient organisms we haven't found, because we see that RNA does not survive for very long. DNA is more stable than RNA under the alkaline conditions due to the absence of something called as the 2-hydroxyl group in the deoxyribose, which makes RNA more prone to hydrolysis. So that's why just remember that there is this 2-hydroxyl group that we have in RNA, but it's absent in the DNA, and this basically leads to easy hydrolysis of RNA. So that's why in moist conditions, in alkaline conditions, it cannot really last for very long, but when it is in dry conditions, it actually can survive longer. So that is what we have seen. Then coming to the next question, consider the following statements regarding mRNA or messenger RNA. Why exactly this question? Because of the Nobel Prize 2023. The Nobel Prize 2023 has been awarded for the work in mRNA vaccines. Now here we are talking about mRNA. So first thing, when it comes to mRNA, we know that mRNA is messenger RNA. Alright, so mRNA is synthesized by the nucleus of eukaryotic cells, this is correct, and then transported to the cytoplasm where it directs protein synthesis. This is fine, because they are called as messenger RNA. mRNA, they are called as messenger RNA. Why are they called as messenger RNA? Because they carry the instructions or messages. They carry the messages or the instructions for making proteins. So that's why this is correct. The process of mRNA synthesis from a DNA template is called as translation. Now let me first also talk about the mRNA vaccines, because mRNA vaccines have been in the news because of this. They use the mRNA to carry the genetic information and prompt the immune system to produce antibodies without exposing it to the actual virus. And these vaccines, they introduce the genetic material corresponding to the viral protein, and typically something which is also called as the spike protein, which is found on the virus's membrane. Now this mRNA vaccine, they are said to be safer because they contain only the necessary elements for the protein expression, and they do not use any of the live viruses. That is the case in the live attenuated vaccines. Now the concept of using RNA therapeutically, it actually began back in 1989 itself, where mRNA being proposed as a vaccine platform for a few years, and then we see that few years later, due to its safety and advantages, we see that we start using it. The COVID-19 pandemic significantly accelerated the development of the refinement of the mRNA vaccine technology. Overall, what we have seen is that mRNA vaccines, they differ from the technologically from the traditional vaccines and the DNA-based vaccines, in that they use a lipid layer to protect the fragile mRNA. When it comes to any viral vector vaccine, for example, something like, let's say, the Sputnik vaccine or the live attenuated vaccines like the Covishield, which can use DNA which are wrapped in a virus, mRNA vaccine, they use genetic instructions which are wrapped in lipids. Alright, genetic instructions, they are wrapped here in lipids. Now the mRNA vaccines, they must be also stored at very low, extremely low temperatures. but their genetic code can be quickly updated for new virus variants as well in some of the other diseases. So that's why we see that mRNA vaccines, the ones for example what Moderna or Pfizer etc. they have used, they have been successful in producing spike proteins and in promoting or rather prompting the immune response in our immune system. So that's why we see that they actually have been very crucial. So coming back to what we are discussing here about mRNA, first mRNA is synthesized in the nucleus and then it is transported to the cytoplasm. So that's why the first statement is correct. The second statement is not correct. The process of mRNA synthesis from a DNA template is called as transcription and not translation. Alright, it is called as transcription. It is called as transcription, not translation. Translation is another process. Translation is the process where you are inserting DNA inside an organism. So that is different. So that's the second statement is wrong. The third statement, mRNA molecules are typically long-lived and they remain stable within the cell for extended periods. Now what we discussed in the last question also is that RNA molecules, they are generally short-lived and they very rapidly they can degrade and they have I mean after they have done the protein synthesis, after that they do not live for very long unless you have extremely dry conditions. So that's why this is wrong, that they are typically not long-lived, typically they are short-lived. So the correct answer will be 1 only. Moving on, question number 18. What are paramutations? So paramutations, they actually refer to interactions between two alleles of a gene where one allele induces a heritable change in the other allele without altering the DNA sequence. Understand this, that they basically one of the alleles of a gene or character, you can say, one allele of a gene, it can induce heritable change in another allele, in the other allele without altering the DNA sequence. Alright, so changes are being brought in without altering the DNA changes, and this is exactly what's happening here, without altering the DNA sequencing. So if you look at the second statement, interactions where one allele induces heritable change in the expression of other allele without altering the DNA sequence, this is correct. Now this phenomena basically results in the modification of the allele having altered the expression that can be passed on to the future generations. Now paramutation is, you can say, in a manner a process which is significant in understanding the gene regulation and inheritance as well. It challenges the traditional inheritance patterns which have been showing that the gene expression can be influenced by the expression between the alleles in ways that do not involve changes to the DNA sequence itself. So that's why otherwise our beliefs or the traditional findings have been different, and where we see that paramutations, they actually have been breaking the traditional understandings. Now in this context, there is another question about PV interacting RNA or piRNA. So the question says, what is the primary role of piRNA? Now when it comes to the PV interacting RNA or piRNA, they also have been in the news in the context of the unusual mutations that protect humans from viruses. Now they are typically type of small RNA molecules that are found mainly in the animal reproductive cells like the sperm and the egg cells. So these tiny RNA pieces, they help in protecting the genetic material in these kind of cells by silencing the jumping genes, which are also called as transposons. They are also called as transposons. So these transposons, which you also in a manner you can say that these are the jumping genes. These are the jumping genes. So they basically will be silenced. Alright, they will be silenced. They will be silenced with the help of the piRNA. Now these transposons, they actually can move around in the genome and they can potentially cause harmful mutations. So by stopping these transposons, piRNA, they help in keeping the genetic information stable and intact, and they ensure that it can be passed on correctly to the next generation as well. So that's why the piRNA cluster, they become very special kind of gene that doesn't make proteins. Instead, it produces small pieces of RNA which are called as piRNA, which are, you can say, small. They are just 25 to 30 bases long. And these piRNAs, they actually work with the PV proteins. That's why the name. They work with the PV proteins, which are which basically help in formation of complexes that search for and destroy the RNA or the DNA with the same sequence as the piRNA. So this system basically acts like a virus trap. All right, this kind of a system that we have, it acts like a virus trap. It acts like a virus trap. So if a virus infects a cell and its DNA, if its DNA integrates into a piRNA cluster, the cluster can produce piRNAs to identify and destroy the virus's DNA, and that's why they will be able to protect the cell from the infection. So that's why if you look at the options given, the second option here, to silence the transposable elements in the germ cell, is what the role of piRNA would be. So as I told you that biotechnology related questions many a times are based on these kind of unknown words. So talking about unknown words, here is another question that we have. What is Casgevy or hexagamglogene autotem cell? So let's try to understand what exactly they are and how exactly do we use them. So now when it comes to hexagamglogene autotem cell, they basically are marketed under the brand which is called as Casgevy, and this is basically a gene therapy that has been developed by CRISPR Therapeutics and also Vertex Pharmaceuticals. These are the two companies involved, and they have been done for treating the sickle cell disease and also transfusion dependent beta thalassemia. So these two, so that's why sickle cell disease can be treated, and also at the same time transfusion dependent beta thalassemia. All right, beta thalassemia. So that's why we see that they basically these are the things that can actually be treated with the help of this new technique. So this therapy, it uses the CRISPR-Cas9 gene editing technology to try and modify the patient's own stem cell in a manner to increase the production of the fetal hemoglobin. So in this manner, what it does is that it improves the function of the red blood cells altogether. So overall, that's why it is becoming helpful, and that's why we see that this also has been in the news that the era of CRISPR therapeutics is here. What can we expect? So this basically is what we are talking about, that until now we have been looking at CRISPR-Cas9 as a gene editing tool where genetic engineering has become very easy with the help of this particular tool. But at the same time, what we see is that now we are going one step ahead and also trying to use them as a therapeutic agent, trying to use them in medical therapies where they can use, or they can be used to produce new red blood cells, and by producing these new red blood cells, they'll be able to solve many of the problems which have been unsolvable until now. So they can solve the problem of sickle cell disease and transfusion dependent beta thalassemia. So this is exactly what we are talking about. So some of these questions can be very difficult, but at the same time they have been in the news. So it's imperative that we should know about this. So this brings us to the end of this particular session. Thank you very much for being here.